Bismillahirrahmanirrahim. Assalamualaikum. Welcome to our microbiodiversity and ecology. So today I'm going to give a lecture on chapter 3, which is on bacteria only. So as you can still remember, we've gone through a bit on bacteria on the previous chapter, so hopefully this chapter will be very easy for you. So first of all, as usual, we're going to have a look at the taxonomy and the nomenclature of the bacteria, and again we've gone through this, the taxonomy, the nomenclature, so hopefully it will be easy peasy for you. So taxonomy, the definition is the orderly classification and grouping of the organisms into categories. And if you can still remember, the eight system of classification, which starts with kingdom and ends with species. And you have to remember that family, it has to start with Asciae, it has to end with Asciae. The genus, it can be referred as the human last name, meanwhile the species can be referred as the human first name. So for example, if a person name is called Ali bin Abu, so Ali is the species name and Abu is the genus name. So the family names, it has to be capitalized, ending with the Asciae. The genus names has to be capitalized, and when we write it in print, okay, so we have to italicize it. Saya ingat kan, when we write it on a piece of paper, the genus and species, we have to underline it, because on paper, tak boleh nak saya ingatkan kan. Okay, for species name, for example, the Staphylococcus sp, we can use this type of writing, using the sp, when we are referring it to the genus only, and when the species is not identified. So there are differences between SP and SPP, where the SP is in the singular form and SPP is in the plural form. We can use abbreviations, for example, Staphylococcus aureus, and it is a very long name, you don't want to waste your time writing it, so you can use S. aureus. So you can just abbreviate the genus name but not the species name. So there are several major characteristics used in identifying or classifying the bacteria. So we can have a look, or we can classify the bacteria through their morphological characteristics, or the physiological and metabolic characteristics, or the ecological characteristics, or the genetic analysis and molecular characteristics. These are the examples for the morphological characteristics, things that we can see. These are the characteristics for the physiological, metabolic, and ecological characteristics. So physiology, for example, the photosynthetic pigments, okay, or the growth temperature, optimum, and range. So for metabolic is how they grow. So is the carbon, they need the carbon and nitrogen sources or not. Okay. The ecological characteristics, whether they are tolerant or they are susceptible to the salt requirements in the ecology. Meanwhile, for the genetic and molecular analysis, in the modern world, we can use a lot of genetic and molecular analysis. For example, we can use the hybridization and the gene characterization, such as the sequencing, to identify and to classify the bacteria. So usually in the laboratory, we can use this identification in order to find the cure for the AIDS. So now we're going to have a look at the isolation, the identification, and the staining. So step 1. We have to strip the culture plates first, Then after we streak it, we can incubate it in. Usually it takes at least 24 hours for the bacteria or the colonies to grow. And for that, we can see the size, the texture, and the color, and we can see if the bacteria need oxygen or no. It is an aerobic or anaerobic respiration. Then the colonies gram stain. This is to observe the cells under the microscope, and we know there are two types of bacteria, which is the gram-positive and the gram-negative. So the main difference between the gram-positive and the gram-negative is the peptidoglycan layer. Okay, so this one is the peptidoglycan layer, the one on the with the violet or purple color. Okay, so we can see here that gram-positive has a thicker peptidoglycan layer in contrast to the gram-negative, which has slightly thinner peptidoglycan layer. So this is a simplified procedure of how to gram stain the bacteria. First, we have to heat or dry the slide first, and then stain it with the crystal violet, then fix it with the iodine, destain with the alcohol, and finally stain it with the safranin. Inshallah, we'll be doing the gram staining procedure in our lab. So after the gram staining procedure, we can have a look at the bacteria under the microscope. So these are the examples of the gram-positive of cocci in clusters. So we know cocci, the shape is in sphere, okay, bulat-bulat. So we can have a look at here is the cocci in clusters, and down here is the gram-negative of bacilli. So bacilli we know is in the rod shape. Okay. So besides the gram staining, we also have the acid-fast staining, where it stains the bacteria with a high lipid and wax content in their cell wall. So for acid-fast bacteria, if we use gram staining, we might not be able to observe it under the microscope. Besides gram staining, acid-fast staining, we also have the acridine orange, where it stains the nucleic acid of both gram-positive and gram-negative, and then we can use to locate bacteria in blood cultures and other specimens where background material obscures the gram stains. Then we also have the methylene blue. So if you use the methylene blue, it will stain the cells blue, and lactophenol cotton blue and chalcoflower white, it is to stain the fungi. India ink. It is an ink or a type of stain to look for the capsules, and in particular for yeast. So if we can look here, the arrow shows for the yeast cells, and with the white halo area, that's the capsules. And by using India ink, we can observe these cells like this. Because some bacteria, they produce a capsule. Why they produce a capsule? Because it is to protect themselves from phagocytosis. So capsule, when we stain it, it does not stain. So it appears like this, like the one in the picture, as a clear area, or we call it as the halo light. It is quite the same if we do it for the antibiotic testing. Besides producing a capsule, bacteria also produce slime layers. Again, it's the same thing to inhibit the phagocytosis. So the microscopic shapes, we have cocci, which is spherical, bacilli is for rod shape, spirochetes is for helical, and for groupings, we have single, pair, cluster, chain, and pedicellin. And we can have a look at the size and length as well. Besides the shapes and the groupings, we can have a look at the size and length. Is it a short or long or curved? Cell appendages. Also, we can use this characteristic to identify, to group each bacteria. And for this, we can see that flagella and pili, we can use this characteristic to classify. So monopolar flagellation is when the bacteria or the bacterium has one side of the flagella only. Bipolar flagellation, two sides of the flagella, and peritrichous flagellation, it means that all over the cell. Okay. So this picture shows a normal and typical culture lab bench where you should have plates, you should have test tube, and you should have hazard bin, and so forth. So this is very typical. Next, other than physiological and morphological testing to identify and classify the bacteria, there is another testing that we can use, which is called the antibiotic susceptibility, to see if the bacteria is susceptible to the antibiotic or not. Okay, so if the bacteria is susceptible to the antibiotic, it will produce a halo around the antibiotic piece. If the bacteria is not susceptible, or in other words is resistant, dia tak kisah pun antibiotik tu, so it can grow even near and under the antibiotic. So we can say that the bacterium is not susceptible, or the bacterium is resistant to the selected antibiotic. Okay, we're going to continue with this in the next part.